Okay, good afternoon, everyone. Could you please tell me if you can see and hear me well? We start in a minute. Okay, sounds ok, I see in the chat. So I also admit some other participants. Cool. Okay, well, while today's participants still connecting to our broadcast, let me say a couple of words as an introduction. Let me just, okay, it's recording is on already. Well, good afternoon, everyone, once again. My name is Matvey Bulat. On behalf of the CNBR Open team, I'm glad to welcome you to the next seminar devoted to the artificial sensations via spinal cord Neuralink. Well, while today's participants connect, let me say a couple of words about the CNBR Open for those who participate in our seminars for the first time. Well, the CNBR Open project unites people who are interested in or engaged in neuroscience and neurotechnology. The name of the seminar may seem, or the project may seem a little strange, but CNBR stands for the Center for Neurobiology and Brain Restoration of the Skolkovo Institute of Science and Technologies. This is a young and modern research center in Moscow. There is where the project was born and is working for more than a year. And the CNBR Open project goals are pretty simple. To allow students and enthusiasts to learn and interact with professionals, professors, and scientists working in different fields. And in order to look after a laboratory or establish contact or just learn from them. Another goal is to connect business and academia, because some of the participants of our seminar are representatives of international companies, and they are sometimes seeking for either inspiration or a project or a solution for developing their businesses. And the third goal is rallying neuro community in which it is interesting to communicate on professional topics and probably collaboratively work on some projects. In this sense, we actively collaborate with the international community, such as Neurotech X, yeah, we have Neurotech X MSK actively working in Russia and for Russian speaking, and we make seminars both in Russian and in English. So I'm really glad to see today some people from Neurotech X among our participants. So hope we'll continue and we will try to do more seminars in English. Well, this is, I would say, the most important I wanted to say. Besides open seminars, of course, we also meet in online and offline neurobars in different cities at the conference events. So just if you want to follow, if you want to join other events, just follow CNBR Open project website, and you can subscribe for updates and news. And now I see that we have participants joining, so let me just introduce Mikhail. Yeah, love the neurobars, Brian says. Me too. Let me ask Professor and Dr. Mikhail Lebedev, the director of the Center for Neurobiology and Brain Restoration at Skoltech, and world-known renowned scientist, and one of the pioneer of the brain-computer interfacing field. So here's him. Let me give him the mic, and Mikhail, I ask you to introduce our today's speaker, Amol Yadav.
Hello, everybody. So it is my great pleasure today to introduce our today's speaker, Amol Yadav. He is a young scientist and a rising star in neuroscience. So he got his PhD from Duke University, where he worked on implanting electrodes into the spinal cord First, he implanted such electrodes in rats, and he started with a Parkinson's disease model in rats, and did really amazing things, like a rat would be made Parkinsonian by an injection of some chemical, and then Amol stimulates the spinal cord of this rat, and then all the symptoms of Parkinson's disease are gone, and the rat starts to move around. Actually, this kind of treatment was also effective in long term. So next, Amol worked on another model in rats. It was an epilepsy model in rats. Again, rats were made epileptic by some chemical. And again, spinal cord stimulation disrupted these epileptic seizures and decreased the number. So this result has a clear relevance to human treatment and human medicine. Next, Amol used spinal cord stimulation in rats to introduce some artificial sensations. So like he stimulates the spinal cord, and this is used as a sensory stimulus, and so this worked great. And Amol even connected like one rat to another rat, and so it in a model called brain-to-brain interface. So after these successful results, he switched to stimulating monkeys. Again, implanted electrodes in monkeys, stimulation. And it appeared that for monkeys, this artificial sensation really worked, and really worked great. So he just recently published this paper, and it is going to become a very prominent paper. And naturally, the next step was to shift to human work. And Amol worked at the Department of Neurosurgery at Duke. And with implanted electrodes, he could restore locomotion in paralyzed humans. So again, success. And now Amol is at Indiana University. He's a professor there, and he is continuing this work there. So hopefully we will collaborate, and this will be a very fruitful collaboration for us all. So welcome our today's speaker, Amol Yadav.
Amol, could you please turn on your video and mic?
Hi.
Okay, yeah, now we can see. Just one word. We have not so many participants for today, so let's probably do the same. After the lecture, we allow people to ask questions orally using their own mic, if you don't mind so much.
Okay. Are you able to see my screen and hear me?
We can't see. I think I can see your screen so far, but I can hear well and see you well.
Okay, great.
No screen yet. No screen.
Oh, no screen?
Yeah, could you please try to share it one more time?
Yeah.
Yeah, it's fine now.
Okay. Okay, yeah. So thanks a lot for inviting me to this talk, and I'm really honored to have this opportunity to share my results with all my friends in Russia and anyone else who is joining from around the world or from the US. And so I do want to say that I really would have loved to be there in person and meet you all, but it's the time of we can now accomplish this task over Zoom. So it's fascinating to be able to connect with all of you from Indiana. So today I'm going to share results from a paper that was published this year, and some more results which are unpublished that will talk about Parkinson's disease and how spinal cord stimulation can be used to treat some of those symptoms. And so to start my talk, first I want to just say that the sensory motor pathway between the brain and the spinal cord is very well understood. So we have the sensory information which travels from the periphery to the spinal cord, and from there it reaches higher brain areas via the dorsal nuclei and then the sensory thalamus and the sensory nuclei of the thalamus, and then from there it reaches the somatosensory cortex. And so from there it's processed, and that's how we generate movements where the signals are now then transmitted back to the spinal cord. And any injuries along this pathway, either in the periphery or at the spinal level or in the brain, can disrupt this sensory motor closed loop system. And so we have a field called the brain machine interfaces, where the idea is that if there's any disruption along the path, either at the spinal level, then we can create a direct interface between the brain and an external actuator or a prosthetic limb in order to accomplish motor tasks. And so the basic idea is that you record neuronal activity from the brain, and you create computational models of movement, and you use those neuronal activity to predict movements and control robotic devices. And so this field emerged about 25 years ago, and in the last 20 years, there's been a lot of progress. So some of the earlier work was done in the Nicolelis lab, where a monkey was trained to control a robotic cursor using its neuronal activity from the motor cortex. And it was taught to not only control a robotic arm, but also to grip different objects. And so in the last 20 years, we have seen a lot of progress in this area of research, where now human subjects are able to control a cursor on a tablet to either play a piano, or they are also able to have bimanual control. So if you've noticed in these examples, the subjects are able to control an external device just by their brain activity. And if you've noticed closely, the only feedback they are getting in this examples was visual feedback of the task that they are performing. And so there's no direct sensory feedback reaching their brain. And so there have been many studies in the last 6 to 7 years, where different researchers have tried different locations along the somatosensory pathway to use those locations for sensory feedback. So, for example, if you notice in the Michera lab, they use peripheral nerve stimulation to encode sensory signals back to the brain. And then this work from University of Pittsburgh, where they're using direct intracortical stimulation of the somatosensory cortex to encode perceptions of touch and even sometimes proprioception. And so what led to my work was I wanted to develop a new technique that was less invasive than direct intracortical stimulation, but also had more specificity and more robustness than, let's say, peripheral nerve stimulation. or have the opportunity to target many locations simultaneously along the body. So that's how this area of research emerged, where we wanted to use the spinal cord as a novel sensory channel to the brain. And the idea was initially to stimulate the spinal cord and then use that as a sensory feedback method in a brain machine interface. But that has led to many different approaches that I've been working on. So one of the aims was to create artificial sensations, so you can use that as feedback in a brain machine interface task. And then the other aim was, let's say if you're able to send signals back to the brain, then can you actually treat neurological disorders, for example, disorders like Parkinson's disease that emerge in the brain. And so, yeah, first I'll talk about the artificial sensations research, and then I will go into using that to treat other neurological disorders. And so we all know that somatosensation is very important. It enables us to perceive the world. It allows us to not only feel tactile and proprioception, but also we can then share our experiences with everyone. And this is how we use to understand the world around us, and that's how we learn how to make movements by using this somatosensation signals that reach our brain. And so without somatosensation, we wouldn't be able to play complex musical instruments like the sitar, or we won't be able to share what we are perceiving with other human beings. And so we really realize how important somatosensation is when we actually lose the ability to perceive touch or proprioception. So for example, this patient here is not able to perform a very simple task of touching, bringing her fingertip to the nose. Or for example, this patient who has to use visual signals to compensate for the loss of proprioception that he has suffered from. So you can see he's focusing on his legs to see where he's going. And so as we know that proprioception is or somatosensation is generally very important, we decided to do a task where we could ask rodents and primates, can you actually perceive sensations that we are generating using electrical stimulation? And so this is a very common task that is used in a lot of neuroscience, where you give animals two options and they choose one versus the other to either communicate that they can perceive or detect sensations, or they can discriminate. So in this paper, which was published in May, we implanted rhesus monkeys with epidural electrodes in the thoracic, lower thoracic, mid thoracic spinal cord level. And similarly, the rats were also implanted with flat electrodes in the thoracic vertebra. And the electrodes we used for primates are the commercially available electrodes, which are used for pain therapy in humans. And so this is a radiograph of the electrodes in their epidural space. And then we had those electrodes come out from their skin, where they were enclosed in a plastic cap, and so that allowed us to engage with those electrodes on a daily basis, and we could connect them to an external stimulator and stimulate those animals. And in the rodent surgery, we implanted electrodes in the epidural space, which is a technique that I've been using for the last 6-7 years, right from the time of my graduate studies. And the electrodes are routed to the skull, where they are mounted and connected to a connector. And so the two alternative four choice task is basically this is how it works. The monkey has to bring a cursor in the center target, and then there are two targets that appear on the periphery. And then depending on whether the stimulus was present or not present, the monkey has to either make a movement of the cursor to the left or to the right to indicate that it can perceive the sensation. So here's a short video of a monkey performing the task in this was collected in the Nicolelis lab. And you can see that there's a monkey sitting in the primate chair and it's moving its joystick. And you can see that stimulation delivered on this side. So whenever you see stimulation, the monkey will make the left movement. So the monkeys learn this task fairly quickly. It takes them about 10 days to learn the task. And so we trained 3 different monkeys on this simple detection task. And you can see that in about 15 days, all 3 monkeys had learned the detection, which can be seen from this curves. And then once they learn the detection task, what we do is we randomize the amplitude that is delivered to their spinal cord, and that allows us to create this graph, the psychometric curve, and then we use that to determine what is the sensory threshold. And so we, based on all our different electrode combinations that we use and the 2 monkeys data that I'm showing here, we saw that there was a huge range in the stimulation amplitudes, which could be considered threshold. And we were able to generate sensations at different locations in their trunk and back and the stomach front body areas. And those sensations were quite stable throughout the whole training period. We would check them very routinely before starting the experiment. And so similarly, we had 2 alternative 4-choice tasks for rodents where they were in this behavior chamber, and similar to the monkeys, they have 2 reward ports where they can choose to go to. So if they pursue sensation, then they're supposed to go to a left port, or not pursue, then they have to go to the right port to get a water reward. So we can train them using reinforcement learning just the same way we can train the rhesus monkeys. So it's a short video demonstrating the rat performing the task. These are the wires that are then sent to the spinal cord, and they get stimulated, and they can get the reward if they perform a correct choice. Just wanted to check that you can hear me correctly and you can see the videos.
Yeah, everything works well, except of there is no sound in the video, but I think it's okay.
Okay. Yeah. I think there's some setting about that in Zoom, but I think we can skip that. Okay, so yeah, so similarly in the rodents, we did the same task where if they pursue sensation, then they go to the left port, and not, then they go to the right port. And we are able to train the the rats on this task fairly quickly. In some animals, it can take about 20 days. Some animals learn even in 10 days. And so moving on, then just as I explained earlier, this is how the perception of the detection of the sensation works. So at a lower stimulus, the animals are not able to detect sensations. At a slightly higher stimulus, they start perceiving them, and then it reaches a plateau at certain amplitude or at certain level where they can always perceive that. And so once you build this psychometric curve, you typically the 75% point on that curve is considered as a perception threshold. And so now the question is like, there are different parameters that determine what an electrical stimulation pattern looks like. And so we can play with, let's say, the frequency of stimulation, we can change the pulse width, and we can also change the duration. And all these parameters are connected to how much charge that is delivered at the electrodes. And so what we did is in rodents, we have the different frequencies. And so we keep the other parameters constant, which is a pulse width and duration. And then we only change the frequency along with the amplitude. And you can see that as the frequency was increased from 10 hertz to up to 500 hertz, those curves, you can see they are shifting to the left. And what that means is that as the frequency increases, if you try to look at the thresholds, then the thresholds are decreasing. And frequency is a very important parameter for sensory neuroprosthetics, and it has been shown that it can be varied to give different perceptions to human subjects or in monkeys in intracortical stimulation. That has been shown that it's a very important parameter. So this allows us to then decide what is the threshold for a certain frequency, and then how can we use this in future in a real sensory neuroprosthetic. And similarly, we also changed the pulse width. The pulse width is also a very important parameter because it controls how much charge is delivered by each pulse. And so what we did is we changed the pulse width from 50 microseconds to up to 400 microseconds. And you can see that similarly, the curves were shifting to the left when the pulse width was increased. And we observed that the thresholds were decreasing. Okay, and so similarly, we also changed the duration of stimulation. And so we started with 50 milliseconds and went up to 1000 milliseconds, which is 1 second. And similarly, we saw that when the duration was increased, the perceptual thresholds were decreasing in the animals. Okay, and then we simultaneously were also working in monkeys, and we were testing the same changes in the stimulation parameters in the monkeys, and we saw that just like I showed in the rodents, as the pulse width, frequency, duration was increased, the sensory perception thresholds decreased in all the monkeys that we had implanted. And so this is data from a single monkey here. And duration and frequency have a relationship with each other. And so what we did is in one animal, we were able to change both the duration and frequency simultaneously in the same experimental session. And we were able to, if you can see in this graph, at a really high frequency, which is about 1000 hertz, we were able to train the monkey, or not train, we were able to see that the monkey can detect the perceptions at really low duration. So at 1000 hertz, even just 2 pulses were enough for the monkey to start sensing those perceptions that we are generating. And so the same results were observed in the monkeys, where as the pulse width, frequency, duration, and frequency and duration together were increased, the thresholds were seen falling down. So once we did the psychophysical testing for sensation for detection, we wanted to see, okay, can the monkeys and the rats understand discrimination? Can they learn to discriminate between, let's say, 2 different sensory perceptions? And so in this, in the same experiment, what we did was we asked the monkeys to discriminate between the sensations, and they did that by using the same task where they would move their joystick to one direction for, let's say, a lower frequency, and then another direction for a high frequency stimulus. So here you can see that both the monkeys learned to discriminate between low and high frequency, which was 100 and 200 hertz, fairly quickly. And then we also did spatial discrimination. So in this, we used 2 different electrode combinations as the target stimulus. And let's say if the electrode between T6 and T7 was stimulated, then the monkey had to move the cursor to one direction. And if T8 electrode was stimulated, they had to move it to the other direction. And the monkeys learned that also fairly quickly in about 10 days almost. So what that means is the monkeys can perceive the differences in the spatial and the temporal patterns of the stimulation. and which is really fascinating to know that they can do that, because then that can really be used in a brain-machine interface task to encode sensory signals back to the brain. And so similarly, we wanted to test that also in rats. And so we did the same experiment where if it was a high frequency stim, then the rats chose one port. And if it was low frequency, they chose the other. And they learned that also fairly quickly. In about 2 to 3 weeks, most of the animals learned to do that. The next thing we wanted to test was this important concept called Weber's law. And so what Weber's law states is that there is a very clear relationship between the intensity or the amplitude that, or any parameter, for example, amplitude that you use to create, let's say, a sensory perception. Then if the animal needs to discriminate between that and some other amplitude, then there is the relationship between the difference that needs to exist between the two stimulus and the original stimulus. So basically, that delta change is directly proportional to the background intensity. And this is better explained with this graphic here, where if you can really see the difference between, let's say, 10 points on a screen versus 20 points, but you won't be able to see the difference between 110 and 120 because it needs to be linearly proportional. So for example, if there were 220 points here, you will be able to see that difference very clearly. And so this property helps us understand, okay, let's say we stimulate the monkey or the rat at 10 hertz, then what should be the difference between that and the other stimulus that the animals can perceive the difference very, very clearly. And so in this experiment, what we did is we stimulated that. We initially trained the rats on sensory discrimination, and then we, in the same experiment, we changed the comparison frequency. So for example, here, the base frequency was 10 hertz, and then the comparison frequency was randomly varied between 20 to 110 hertz. And you can see that as the frequency difference is increasing, the animals are able to perceive the difference better. And so then we repeat the experiment for different base frequencies, which is 20, 40, up to 100 hertz. And we then plot the same curves for each of the data that we collect. And then we consider the 75% mark on each of these curves as the threshold that is needed for perceiving a difference. And so that's plotted here. So let's say the base frequency was increased from 10 hertz to 100 hertz. Then what was the just noticeable difference for each of these frequency values? And we saw that there was this linear relationship between that. And so we were able to assume that the Weber's law holds true for spinal cord stimulation based sensations. And similarly, we also wanted to compare when the animals can perceive a difference between the high frequency and then if the comparison frequency is a lower frequency. So for example, here the base frequency was a higher frequency, and the animals then had to discriminate between that and lower frequencies. And you can see that similarly, there was this relationship as the difference was increasing. They were able to perceive the difference much better. And similarly, there was a linear relationship between the difference and the base frequency. So in conclusion, in this paper, which was published a few months ago, we were able to claim that rats and monkeys are able to detect and discriminate sensory percepts that are generated with spinal cord stimulation. Not only that, the threshold for detection decreases as the frequency parameters such as stimulation parameters such as frequency, pulse width, and duration are increased in both the animals. And then we also were able to prove that the Weber's law holds true for sensory discrimination. And this is very important from the perspective of developing a sensory neuroprosthetic, because if all these questions that we asked about the stimulation parameters are true, just as they are seen in intracortical brain stimulation, then we can very essentially say that we could have an alternative way to provide sensory feedback. And so what are we doing next? Trying to see how to get rid of this. Okay, so next what we want to do is we want to develop high density recording and stimulation from different brain areas as the animals are performing this detection discrimination task. We want to use more basic neuroscience questions like what are the brain circuits involved in this detection and discrimination. And then simultaneously, as we understand that, we also want to test this in human subjects where human subjects with either a spinal cord injury or any other neurological injury where they are not able to perceive sensations. And so we want to understand what are the thresholds for different stimulation parameters. Can they detect and discriminate sensations, and also have a map of the sensory responses? And this is fairly doable because spinal cord stimulation has been used for pain therapy for several decades now. So all we have to do is create this interface with an external stimulator and the leads that are used in patients. So there's a period of about a week or 2 weeks where after getting an implant, the patient's leads are accessible. And so we are planning to do some of the initial testing in those patients who get implanted for pain therapy.
Amol, maybe we ask if there are any questions so far from participants.
Yeah, sure. Yeah, go ahead.
So dear participants, if you have any questions, so probably we can make a short, yeah, there's just unmute yourself. So I think it should be allowed. Yeah. You can also turn on the video so that we can see who's asking.
Yes, do you hear me?
Yeah.
Hi.
Hi.
Hi, everyone. Hi, Amol. It's
Hi.
Giacomo Valle from ETH Zurich. Very nice talk.
Hi.
Very great work. Congratulations. I have a couple of questions about the approach in general. So because I'm working also in the sensory restoration in this field, more with the peripheral nerve stimulation. So I'm very interested about the feasibility of maybe use spinal cord stimulation in other cases. I work mostly with amputees. So the first question is about the target population of patients, for your technology. So can you clarify maybe what type of patient do you see that maybe in the future they can use your technology? In particular, if you consider that in case of amputation, maybe they don't know the risk, or they prefer to have a less invasive implant in the periphery, I don't know, respect to touch the spinal cord. And in case of spinal cord injury patients, maybe the spinal cord sometimes is damaged, it is not an healthy spinal cord, and the placement of the electrode can be difficult.
Yeah, no, that's a really interesting question. And so finding the right patient population to do initial validation and testing is really crucial. So certainly the amputee population could be a good target, and there are other labs who have already started investigating that, and they had a publication on that topic already from the University of Pittsburgh. And I think from spinal cord injury perspective, there are patients who who undergo spinal procedures like corrective surgeries after their injury. And so those patients could be a good target because as long as above the lesion, the spinal cord is intact, you can stimulate that area and be able to send this signals to the brain, and then they can perceive. So there is a way to use spinal cord injured patients also to do validation of this.
Thanks. If I can,
Yeah,
maybe
sure.
another.
Yeah.
Thanks. The other question is more about the technology. So I know also exactly reading the paper published by the group of Pittsburgh, Gaunt and and the others, and Lee Fisher, I saw some problem regarding the migration of these leads, after the implantation, for the movement, of the, and they don't know still if, for example, there is also some migration due to the neck movements or movements in general. So did you observe the same in your case, or?
Yeah, no, definitely. This is the migration of leads has been an issue, not just in this investigation, but also for pain therapy for several years that spinal cord stimulation has been used. So the way the current leads are implanted, they are just sitting on the epidural surface and they can move around, not so much, but they definitely can move around. And so there are studies where, depending on the posture of the patient, the distance between the spinal cord and the epidural space can shrink or enlarge, and so the electrodes contact can change, and so the impedance changes, and so the amplitudes, the thresholds change often. So I think a solution to that could be to create high density electrodes that can cover the spinal surface just like an ECOG grid. And so that could be something future technology. And so that's one of the aspects that my lab is working on right now, is to create this high density electrode grids so that there's not much movement in the rostrocaudal or lateral space. So that could be a solution, and that will be a solution for a lot of different disorders where spinal cord stimulation is used.
Great, thank you very much.
Thank you, Giacomo, for good questions. Maybe there's another question in the chat, but probably we'll proceed with the talk and then ask this question at the end, if you don't mind.
Okay, yeah, just let me know if I need to stop or answer in the middle.
Yeah, if anyone has a question, just raise their hand so we know, so I know that, yeah.
Okay, so let's move on. So the idea would be to implant the subjects and then ask them similar questions that we were asking the monkeys, but instead of here we can actually ask them very direct questions like, can they perceive or not, and what type of sensations are they perceiving with this electrical stimulation, and then how natural those perceptions are. And so that's something that I want to work on, and then the long-term goal is to create continuous sensory feedback. So either for example, I'm using an example of amputee subjects here, but I think any other patient population, this could be applied. And so in this initial part, I have shown that if you can create a novel sensory channel to the brain, you can send signals and create perceptions and sensations that the animals and humans can perceive. So it has the application to be used as a path for sensory feedback, or to just send sensory signals to the brain if you want to do that, not in the context of sensory feedback. So the next thing would be, can you actually use that to treat neurological disorders? So let's say there's a disorder that emerges in the brain or it associated with the brain. Can you actually use those sensory pathways and the sensory signals to treat those disorders? So in this next part, I will talk about a very well-known neurological disorder, which is Parkinson's disease. And so this was also a line of investigation that was started during my PhD work in the Nicolelis lab. And some of the earlier works on this topic are very well known. So for example, it is very well known that there's this pathological synchronization in the brain of Parkinson's patients, and often it is observed at around the 10 to 25 hertz beta frequency range. And it is very well known that if you stimulate the subthalamic nucleus or the GPI with deep brain electrodes, then you can actually desynchronize this oscillation. So this is a very well known fact in the neuroengineering field or the neural stimulation field. And so some of the earliest work on this topic was done by Ramiro Fuentes, who was a postdoc in the Nicolelis lab. And what he showed was that if you if you stimulate the spinal cord in a very acute mouse model of Parkinson's disease, in these animals, they have depleted the dopamine for a very temporary period. And so you can see that as the stimulation was turned on, the animal was able to move. And before that, it was very frozen and akinetic. So this was the first showed in 2009. And so that really inspired me to start working on this concept. And what I wanted to do was I wanted to create a rodent model which would be more for a lot of the models that I used are unilateral lesion models. So for example, if you inject a drug called the 6-hydroxydopamine, it results in the loss of the dopaminergic terminals in the substantia nigra and also in the striatum. And so this is a progressive model that you can see that over a period of about 3 to 4 weeks, most of the dopaminergic terminals or the dopaminergic neurons in the substantia nigra are destroyed by this neurotoxin. And so similarly in the striatum, you can also see that the dopaminergic terminals that innervate the striatum are lost progressively. And so I wanted to create this bilateral model that allows progressive investigation of stimulation in these animals. And so I injected the drugs in the intrastriatally in the both hemispheres, and this is some of the symptoms that we were able to see in those animals. So this was a normal sham lesion animal. You can see it is moving around in the open field chamber fairly easily. And this was a dopamine depleted model that you can see it as a crouched posture and does not have a proper gait. And so what in these animals, we were able to show these results that if you stimulated the animals in the dorsal column using the epidural electrodes that we design in our lab, the animals that are in this green trace, their symptoms start at the same place of all the three groups. So the red is the Parkinsonian, the green is the Parkinson plus the stimulation, and then the blue is the sham lesion animals. So they all start at the same principal component space, and then the red, which is the animals with the lesion, they kind of deviate in a separate direction, while the green, although it starts in that direction, it starts after a few weeks, it comes back to the sham lesion animals. So this was our way to show that over a long period of time, if you stimulate these animals, they will recover, their symptoms start improving over a period of 6 weeks. And so the hypothesis at that time was that you have these oscillations in the brain, right, in the motor cortex and the striatum, this corticostriatal oscillations at the beta frequency, which have been shown. So maybe we are able to send by stimulating the spinal cord, we are able to send signals to the brain that can desynchronize this oscillation. And so this was a hypothesis that we have been proposing in this paper that was published in 2017. And so if we are able to send signals to the brain and can desynchronize, the next question was, can you do that in a real-time closed loop fashion? So that was the next question that we started investigating, was can you stimulate the spinal cord in a closed loop fashion, where you use the neuronal recording to identify oscillations in the brain and use that to trigger stimulation instead of having it continuously going. Okay, so for this, what we did was the similar work as the previously published paper, where we have electrodes in the two brain areas, which is the motor cortex and the dorsolateral striatum. and the electrode assembly has the option of injecting drugs into the striatum to cause a lesion, and those electrodes are also movable. And then simultaneously, you also had electrodes in the spinal cord. And so the protocol for our closed loop stimulation was we did the surgery, then we create a lesion. So in the surgery, we implant the electrodes both at the spinal and the brain level. Then we create a lesion with the injection of 6-OSDA. We do a pre-lesion recording, then we do a injection with 6-OSDA. And then over the next period of 3 to 4 weeks, we did all different investigations of open loops versus closed loop versus off stimulation sessions in those animals, and then later we perfuse their brain. And so a typical session consists of some baseline recording where we use that to identify either oscillatory activity or some spiking activity that is spiking or LFP activity that can be a biomarker for Parkinson's, and then we use that to trigger stimulation in the spinal cord. And we used some of the center part of the session to do our investigation or analysis. So there are two different approaches we used. One was a spike triggered stimulation. So in that we identify, we record from multiple neurons during the baseline period. And then we identify either 2 or 1 neurons that have an oscillatory activity in the 10 to 25 hertz range. And then we use that neuron's activity to trigger stimulation. So that was one approach that we tried. And then the other approach was the local field potential oscillation. So in that approach, we also, along with the spikes, we're also recording the local field potentials. And then we use some signal processing like Fourier transforms to calculate the power in the beta frequency range. And then in this approach, what we do is every time the power crosses the certain threshold that we have determined, then we stimulate the spinal cord. And so this data is collected during the baseline period, and then that baseline period is used to identify what is the threshold, what LFP to use or which neuron to use to use as a trigger. And this is a very simple demonstration of what's going on during a particular session. So you have the LFPs and you have the envelope of the beta oscillations, and every time there is a threshold that gets crossed, we trigger a train of stimulation to the spinal cord. And that's happening in real time as the animal is either in the behavioral chamber or in the open field, and it can stimulate it immediately. So this is a video of an animal in the open field chamber where you can see that it is very immobile during the off stimulation period. This model is very severe, so that rats are quite kinetic during the after the lesion, and so this closed loop stimulation is turned on, and you can see that the rat certainly will start moving and it will start exploring the open field during the closed loop period. So some of the results that we observed in our investigation was the movement of the animals that we quantified. So you can see here before the lesion, this is a sample session of a rat. This is during the off stimulation period. So this is after the lesion has been performed, and a week after that, I think. This is the off stimulation. This is how the rat was moving in an open loop stimulation, and that's closed loop stimulation. These results are not yet published, but we are working on submitting a paper very soon. Here you can see that the same quantification with pre-lesion, off stimulation, open loop stimulation and closed loop stimulation. So this was quantified in several animals, and we were able to show that there was a very clear improvement between off stimulation and closed loop stimulation in the amount of distance that was traveled, and also the time that the animals spend in the resting phase. You can see that the difference between this green and blue bars is very significant there across all different animals. And then even in we also started looking at what is the effect of stimulation on the oscillations itself. So, like we're using the oscillations to trigger stimulation, but we also wanted to see, does that actually impact the oscillations in return? So that's something we were able to show. We are able to show here that there is a very clear difference between the oscillatory activity in the beta frequency between the off stimulation periods and the closed loop stimulation period. So what that means is not only we can use the stimulation to use the stimulation as a way to be triggered by the oscillation, but also it actually impacts the oscillations in return. And this is something new that we are still investigating, and we are in the process of figuring out. So we wanted to understand, okay, is there a difference between let's say when the animal is initiating movement, because our hypothesis is that we are desynchronizing oscillations in the brain. So do we actually create a state, a neural state that facilitates locomotion? So in this graph, what we have done is we have taken the periods before locomotion and during locomotion, and periods in two conditions. So in this, the orange the ellipse and the points are during the off period, and this blue ones are during the closed loop stimulation period. And we were able to see that there is a clustering that happens between so let me explain. This is this is based on the firing rates of the neurons that are recorded during these two conditions. And then what we do is we do a PCA analysis of the firing rates of the neurons that were binned at different times during the pre-locomotion and post-locomotion period. And you can see this cluster forming pre-locomotion and post-locomotion in the off period, and similarly pre-locomotion and post-locomotion in the on stimulation period. And there is a very clear difference between the proximity of the clusters and also the sizes of these clusters. So we are still trying to understand what this means. I think one way to explain this could be that there is a variability, there is a higher variance in the firing rates of neurons in the pre-locomotion period, and stimulation somehow reduces that variance and creates a state that is pro-locomotion. And in the stimulation period or the stimulation phase, that happens very easily as compared to the off period. And similarly, we also looked at what is the average frequency that we are delivering to these animals, or what is the variation in the stimulation pulses. So in this graph, you can see that in the open loop stimulation, you have constant stimulation at 100 Hertz, but when you do closed loop stimulation, you can provide stimulation on average about 10 to 15 Hertz. And similarly, the coefficient of variation is also very high in the closed loop stimulation. And that is sometimes important in terms of creating stimulation patterns that are directly addressing the oscillations, because not only you want to reduce the charge that you're delivering to the spinal cord, but you also want it to be very precise in its timing so that you are targeting the oscillations, right when they happen. So with our protocol, we are not only able to reduce the amount of charge that we are delivering, but we are also creating more variation in the patterns so that they can be, be more precise in targeting or desynchronizing the oscillations, as compared to constant open loop stimulation, which is always on at a certain frequency. So in conclusion of that, those results, we can say that closed loop stimulation of the spinal cord improves the motor function in the Parkinsonian rats. And closed loop stimulation not only reduces the oscillations in the striatal areas, but it also creates this locomotion or pro-locomotion state, which helps the animals to then initiate locomotion. And it reduces the amount of charge that is getting delivered and as compared to open loop stimulation. So in terms of some future directions and clinical translation, we want to figure out what is the more optimal way to stimulate the spinal cord in these rodent models. I'm sure there's a relationship between different stimulation parameters and desynchronizing of these oscillations in a very precise way. So we want to investigate that in the future. We also want to see if simultaneous deep brain stimulation or spinal cord stimulation can have a better impact in rodent models. And in terms of clinical studies, we want to optimize the parameters to treat Parkinsonian gait disorders such as freezing of gait. And then we also want to create the closed loop stimulation in human subjects. In terms of some technological improvement, like I mentioned earlier in the question that I was asked, so in our initial work, the very early work, we used 2 contact electrodes in the spinal cord. More recently, in the published paper, we were able to go up to 4 contacts, and now in the future work, we want to use high density micro contacts, maybe up to 32 to 64 channels, so that we can not only create very precise sensations from the sensory neuroprosthetic perspective, but we can also, from like treating neurological disorder perspective, create very precise activation of the spinal cord circuits. And so that's something that we are also working on right now and testing it in animals. So in conclusion, I want to say that with the earlier work that I shared, we can claim that spinal cord stimulation is a novel sensory channel, and it has a very robust psychophysical relationship that can be exploited for sensory neuroprosthetic applications. Not only that, if we can actually send signals to the brain, then we are definitely open the possibility of treating neurological disorders that emerge in the brain. And so if we can modulate neuronal activity, let's say for Parkinson's, there is no reason to believe that we may not be able to do that for other neurological disorders. So for example, our approaches can be used for several diseases and disorders. And our goal in our lab is to create platform technologies that can then be employed for different disorders and injuries. And so with that, I want to stop there and acknowledge my mentors and collaborators for the last several years. More importantly, in the Nicol Ellis lab, my PhD advisor and postdoc advisor, Dr. Miguel Nicolelis, and the postdocs that I worked with, postdocs and senior scientists such as Romulo Fuentes, Kafu Iziraza, Eric Thompson, Miguel Vieira, Misha Lebedev that you all are familiar with, and other mentors such as Craig Enriquez and my neurosurgeon, collaborators like Nandan Lad and Max Kukov, and all the students that I have worked with at Duke, and also my current collaborators and mentors here at Indiana University, and the several funding sources that have supported my research in my previous lab and currently as a faculty. So with that, I want to stop and take any further questions or any comments or any suggestions, recommendations. I'm open to discussing this.
Okay. Thank you very much, Amol, for your great presentation. It was very like a lot of things to think on and to see. We have several questions. Some of them are in the chat, and some of them are in oral form. So let's start with those who wanted to ask in person. Could you please just raise a hand and then unmute yourself? There was Tas, as far as I know.
Hello, can you hear me?
Yeah, you can also turn on your video if you'd like to.
Yes, can you see me?
Yeah.
Okay, so first of all, thank you for the lecture. It was a great lecture, very interesting and very inspiring for me. So my name is Stas. I'm a third year bachelor student at MIPT, and I'm really interested in this field of research of sensation recovery and neuroprosthetics. So I would like to ask some general questions. The first one is what are the main subjects, disciplines, or skills maybe that one has to acquire to work in this field of science?
Okay, yeah, that's a very interesting question, and it's great that you are asking this very early on in your career, and it's really inspiring that you are interested in this area of research. And we always, as professors, are excited to get those questions from students. So, in order to participate or be involved in our field of research, I think there's a combination of neuroscience and engineering that's going on. And so we are considered the field of neural engineering, where you learn not only the basic concepts of neuroscience, for example, more of a systems neuroscience, but you also learn engineering principles, for example, electrical stimulation, or how to do computational modeling of different circuits and neuronal populations, or we can also approach this field more in an empirical way, where we can learn how to do surgeries in animals and then build those electrodes, build the computational the apparatus for doing electrical stimulation. So for example, there's a lot of signal processing involved. So how do you record spiking activity or local field potential activity, how to process that in real time to get oscillatory activity or any other information from the activity of the neurons. So there's a combination of a lot of different fields that goes on in our area of research, but primarily the focus would be neuroscience and engineering together. So I think Dr. Mikhail Lebedev would be a great resource to ask in more detail, because he is a really great mentor for me and other people in the Nicolelis lab, and he really told us like exactly what we need to learn to master this field of brain-machine interfaces. So certainly reach out to him, and also feel free to reach out to me with any specific questions about like any research that I presented today, and I'll be happy to share and tell you what path to take.
Okay, so can I contact you maybe via the Gmail or something like that after the lecture, not to waste time on general questions?
Yeah, definitely. You can see my email here. This is my email address. You can send me an email at that.
Okay. Thank you for the question. There is another question in the chat. It's again some kind of general: how to enhance an electrical conductivity of electrode and skin compatibility? How it depends on electrode material. It's from Amrit. He has or she has some mic issues. So I can ask.
Yeah. Okay. So this is not really my area of research. About I think that falls a little bit into biomaterials and different electrode tissue interface research, which people who do more computational modeling, they're addressing that, or people who are more into the building of the electrodes and the materials, they are the best to deal with that. I generally collaborate with others who are experts of that, and then come up with designs in terms of, like, let's say the orientation of the electrodes, or how big or small the contacts need to be to activate the spinal cord circuits, but I would say like I'm not really an expert of the materials that are used.
Okay. Do we have any other questions? Please raise a hand. Yeah, Gurgen, you're welcome.
Yeah. Hi, can you hear me?
Yes.
Yeah, together with some background noise.
Yeah, I'm really sorry I cannot do anything with that noise, but I really wanted to ask a question. Thank you for the great presentation. It's really very interesting. I'm currently a PhD student who is going to work with such a project of sensor restoration with Mikhail Lebedev. And I made some literature review during like last several months. And as far as I see, the whole idea of the sensor restoration is just more about to replace, to change our nerves with something like electrical, with like electrical material, with other materials. But I think that the other part of this sensor restoration can be just to like make our nerves to like grow with some additional effort. And as far as I see, there are some works with like stem cells and scaffolds on rodent models. So my question is, do you do such studies in your lab? And the second one is, how do you think, is it a good idea to try stem cells and scaffolds for like the next level of sensor restoration? Thank you.
Mm-hmm. Yeah, this is an excellent question also. I'm not typically doing research that involves more cellular and molecular based therapies. I'm using purely engineering, like more electrical engineering approaches to treat and restore sensations or motor disorders. But there are other people who are working on this area, and certainly the challenge is often when you implant this new cells or stem cells, then how do you train them to actually behave like the original structures or original, the cells that are lost? So that is still a challenge that people haven't figured out well. So I think that could be an interesting field, and certainly there could be some approaches where electrical engineering based therapy can be mixed with other cellular biomolecular approaches. So that's that's something that you should definitely think about and pursue.
I see. Thank you. I definitely agree with you. Thank you.
Okay, thanks, Gurgen, for your question. There is a raised hand by two guys. So Anna, probably you can.
Can you hear me? Can you see me?
Yes.
Yes. So thank you for good presentation. And I have a little question. Is there any resistance to electric stimulation of spinal cord? And is it necessary to increase the electric current?
Yes, there is a resistance. You're meaning from electrical perspective, right? Not abstract resistance. Okay, yeah. So from a electrical perspective, definitely there's a impedance that impacts the relationship between the electrodes and the spinal cord. And like I mentioned earlier, depending on the posture and the way the patient or the animal is positioned, that resistance often changes. So we did observe that in our monkey studies and in our rodent studies, often the thresholds, they do change. So we have to calibrate our experiment every time we are doing a session, like a stimulation session. We look for what is the perception, what is the threshold, and what is the actual impedance. You can do impedance tests to figure that out. And even in the human subjects, when they are implanting the electrodes, they check for the impedance at the time of the surgery, they do that after 1 week of the surgery. They sometimes, even if they have access to the electrodes, then they even do that in the future. And often the resistance changes because there is all kind of response to the the foreign body that's implanted in the spinal cord. So there's astrocytes and microglia and all, they can have impact on the resistance. So yeah, that is something that needs to be calibrated regularly.
So if I understand it's right, you calibrate individually and at each session for any object. Yeah. Okay, thank you.
Yes.
Thank you.
Thanks, Anna. We have another question from Giacomo. Could you please unmute yourself?
Yes, I'm interested still in the part of the sensory feedback restoration. And I have one question because one thing that we observe in general with the electrical peripheral neurostimulation is that proprioception, since you mentioned before both proprioception and tactile sensations, no, we saw that it's quite difficult to elicit a realistic natural sensation of proprioception, no, in the using electrical stimulation. So also in your study, for example, you report that you are eliciting some somatosensory sensations, that is, but you are not sure about if it's a combination or is only tactile. I mean, or can you comment on how to disentangle proprioception and touch, and in case if it's possible to elicit proprioception also with the spinal cord stimulation? Thanks.
Yeah, definitely. This is a really tough question because this is something that we have been talking since we started this study. Like, how do you separate tactile and proprioception, and can we actually even have a proper task where we can ask the monkey or the rat, like, can they actually pursue proprioception? Because that's always a challenge, because not only they're actively performing a task, so they are generating proprioception constantly while they are doing the task. So how do you even disengage between their active proprioception and we creating some artificial proprioception? So that's a really, I think, a tricky challenge. And I think that the way to address that would be to have more precise targeting of the spinal cord. And for example, we do know that the dorsal columns are conveying mostly like light touch and proprioceptive information. So it might be something that we might have to investigate, either using very precise stimulation or maybe some kind of optical approaches, like optogenetics, to maybe target very specific neurons in the spinal cord or specific pathways that then we could maybe dissociate between the two. But the task itself is also a challenge. Like, what task do you do in an animal that differentiates between the two?
Great, thank you.
Okay, thanks, Giacomo. Do we have any other questions from the audience? Let me check in the chat. Okay, it seems that not anymore. Yeah, as far as
Amol?
there is a question from Mikhail Lebedev. Please, could you please turn on your video so that everyone sees you?
Can I ask a question? Amol,
Yes.
my video is on, I think. Let's see. Can you? Sure. So, Amol, could you speculate if spinal cord stimulation could be helpful somehow for these people somehow, and where would you stimulate for this purpose?
I missed which people?
It seems that the internet connection is not good.
Stroke. My internet connection is bad. People with the stroke.
Okay, yeah. So this, in terms of, okay, people with stroke. Yeah, I think this could be useful. Could be useful for in those patients also. Stroke often can lead to loss in the brain itself. So the question would be, if we do stimulate the spinal cord and the signals can be transmitted to the brain, can there be like other areas that can, be involved in the perception? So I think it would kind of depend on where the stroke is in the brain, and as long as the signals can reach the areas where sensory perception is happening, I'm sure it can be, spinal cord stimulation can be used for restoration. Did that answer? I'm not sure if I missed part of the question.
I'm not sure.
Okay, so it seemed that Mikhail was satisfied with your answer.
No, it was great. Yeah, it was great.
Okay. Well, do we have any other question? We're about to wrap up the session. Yeah, I don't see any other raised hand. So then let me thank you, Amol, one more time.
Actually, one thing I wanted to say in response to Misha's question was, I just remembered something. So, by stimulating the spinal cord, we are actually activating some of the natural somatosensory pathways. This is how the sensory information reaches the brain from the periphery. So there's always a question, like, if you stimulate the brain, are you activating the the neurons in a very specific area directly, which may not be the natural processing, how that happens. So one of the ways we like to think about this technology is that we are incorporating all the filters that exist in the different those somatosensory pathways, and so we are able to use those filters in the circuits and all the synapses that are already embedded in the circuits to create this novel pathway. So it might be possible that it could be more naturalistic, or it might be possible that we don't have to focus on where in the brain the information goes, as long as it can be perceived and it can be learned, or it can lead to plasticity.
Okay, I see the thumb up from Misha. So it's nice. We have another question from Daniel. Daniel, please unmute yourself and turn on the video if you can.
Yes. Hello. Hello. Can you hear me?
Yeah. Go ahead.
So I kind of an accidental listener to this seminar. I actually don't have any research related to the brain stimulation. I wanted to ask if the tools you use, if the chipsets and electrodes are custom, or do you purchase some commercial pre-built hardware?
Okay, yeah, definitely. That's a good question about how we build all the systems. So in terms of the electrodes, in monkeys, we purchase the electrodes commercially from this company called Abbott. They have been manufacturing stimulation electrodes for a long time now. So those are the percutaneous electrodes that are implanted they're cylindrical, and so they go in the epidural space, and they are used for pain therapy. So we just use the ones that are approved, FDA approved for human work. In rats, we build our own electrodes in the lab. And so these are platinum electrodes that are coated in a medical silicone casing that we implant on the spinal surface. And for doing more of the recent work that I'm doing, the high density electrodes, I am working with a commercial partner to design and fabricate them at a commercial location because we don't have the technology to do that in our lab, so we just outsource that to a commercial fabricator.
And talking about
Yeah.
software, do you write it by your own as well, or you just ask your commercial partners to?
No, the software we write our own. So in terms of the neuronal, let's say the brain recordings, there are a lot of companies that are providing the hardware and all the interfaces to connect that with either MATLAB or Python. So for example, there is Plexon that's been around for many decades. Now there is BlackRock, which is about 20 years old. There's a new company called Ripple, which is about 5 to 6 years old. So there are many different commercial companies that are doing this, but there are also different open source approaches. So there is OpenEFIS, or there is Spike something, GLX or something that can be used for doing more open source building of the electrodes or the interfaces for signal processing and amplification and filtering and all that. So there's a different, depending on the budget of the research, you can try different approaches. You can either go to the commercial partner companies, or you can even build most of the things by yourself for a couple $1,000s.
Yeah. And you use the controller which is mounted onto the cage, the controller for the electrodes, or?
It is, yes. So I've used all different types. So for the monkey project, we had a custom design stimulator that was built in the Nicolelis lab by a graduate student long time ago. His name is Tim Hansen. So we used that. He was the one who founded Neuralink, by the way. So we used his stimulator. Then for the rodent work, we have used Master 8 and Master 9, which has been around for many years. And then right now for doing high density work, we are using Ripple Systems stimulator. But if you want to go the more open source way, you can work with Intan, which has stimulators that are not very expensive. But if you also want more expensive, then there's BlackRock and Ripple, which can provide that.
Thank you very much.
Thanks. So do we have any other question? Please raise a hand. Yeah, I think we're done with the questions. And this is a good time to wrap up the session. Amol, thank you one more time for a brilliant talk and for all your answers to all the questions. I hope that next time we see each other in person, either in Moscow or in some other place. Yeah, so waiting for the end of Corona, and then create a meeting with all the interested people in stimulation and brain-machine interfaces. And I would like to thank all the participants of today's talk, of today's seminar, for the enthusiasm and for great questions. And yeah, it was like a real interest, and I'm really glad to see this. And thank you, Mikhail Lebedev, for joining us today and for your participation in this seminar. Subscribe for the news and to know the updates on the seminar. We continue. We also will do some other seminars on stimulating, and so join anytime. See you next time. Thanks, Amol.
Yeah, thanks. Thank you very much, and really enjoyed talking to you and presenting. And yeah, I'd love to meet you all in person, hopefully very soon. And yeah, stay in touch. Send me any questions you have, and if you have ideas that you want to try and test, yeah, feel free to reach out and we can find some way to work.
Okay, great.
Okay.
Bye.
Bye. Bye. Bye.
Bye.